I know, Adi, you'd mentioned at one point too during your discussion, the importance of certain digital tools. You mentioned soft sensors, right? There was, you've done some writing, or I know academia has been exploring the use of digital twins, right?
Yeah.
So that's become another big piece here to help us advance quality by, as you and your colleagues termed it, right? Digital design, QBDD, for more advanced process modeling and control. So how much attention, in your opinion, Adi, is being paid on advancing this today? And how are you seeing these tools being implemented into manufacturing processes today, if at all? And that's for you, Adi, first, but then April and Tyler, if you want to jump in.
I'd say there's a huge push towards digitalizing the overall manufacturing space, right? Because again, bioprocessing 4.0, all of these terms, the fundamental idea is how much information can you get from a manufacturing process? A real process, how much information can you extract from it? And then using that, you make a virtual model of this, an ideal model, right? And then put some constraints for what's the quality that you are looking for as far as your product is concerned, and use that virtual model as a reference standard to make sure that your real process is maintained in a particular stage, right? So basically, it's a control mechanism. Like the final goal would be to have a control mechanism, right, with these digital twins, and a step further would be advanced control mechanisms where, even before your manufacturing process goes into that danger zone, the system already knows, okay, if these events are detected, please do these steps so that it doesn't go to the danger, the red zone, right? So that would be the final goal. And then, I have seen the same transition in not just in the RNA field, in multiple fields where automation has been the main, or one of the main driving forces, right? And if automation is your final goal, then you definitely need some sort of digital tool to control your process. And then that's where all of these, final digital tools, computational tools like soft sensors and digital twins come in. So definitely there's a huge push in achieving or in implementing these techniques as far as RNA manufacturing is concerned.
Tyler and April, I know one of the other questions I had, right? Automation is becoming a bigger buzzword. I think it's every industry before has gone through it, right? Cell therapy, gene therapy world's been talking about it for a couple of years. I'm starting to hear it crop up more at conferences in the RNA space. Can you kind of unpack like where you're seeing some of the biggest gains or shortfalls, right? In automating our processes and tie that into some of these advanced tools, right? Digital twins or soft sensors, right? To help us really understand what's going on and shape our processes in real time. Whichever one of you.
I don't have
April's like, I don't know.
Throw Tyler to the wolves first. No, I completely agree with what Adi was saying. And I think we've had experiences where, operator errors occur, right? So if there's any steps where you can make it automated and you have feedback loops, and if there is in-process monitoring of certain analytes where now the instrument knows, oh, I need to add more buffer, or I need to add this or that, versus an operator getting signal to then add. And you try to build as much a foolproofness into batch records and making sure everything is as tidy as it can be, but there's always that operational error that can happen. It's human error. And so I think that's automation. That's where I really see it in the near term now. And a lot of that, I think, has to just come back to kind of who you choose as your vendor for certain bioreactors or certain downstream processes, and what are those modules, what are those add-ons, what's the software that they're utilizing, and making sure that they're compliant for your GMP processes and ensuring automation.
Yeah. April, what do you think? You agree?
I'm sure. And I think at the screening stage, it's important to consider the automation approach because when you're flying through a lot of different iterations of mRNA and they have to all be plate-based and we're screening tons of different things. The purity of them, how they're made, and how that eventually relates to something that you're going to put into, say, animals or later on humans, becomes an important question. So you've got automation at the high throughput stage, but also automation later, and it's how they relate to each other that should be considered. Like right now, I think the automation we do at the high throughput stage is not going to achieve the sort of representativeness that one might wish to move into later stage studies. But it's a necessary tool. So the better we can align those and start trying to incorporate as much of the analytics that we're discussing here into that high throughput approach, the better. I mean, we're talking small sample sizes, the analytics are going to be limited, but at least getting enough knowledge to be able to optimize it and have it be predictive. I mean, that's always my gripe with high throughput systems is it going to be predictive of how it behaves in vivo and after we've made it in a manufacturable way, because there can't be changes. So.